This is Biochemistry Unfolded, from atoms to algorithms, a weekly podcast on the topic of computational biochemistry and biophysics. Hey, good afternoon. Welcome back to another episode of Biochemistry Unfolded, from atoms to algorithms. So in previous episodes, I've talked a lot about artificial intelligence, deep learning, and the role this new technology takes in playing out in the science of biochemistry and protein structure prediction, as well as drug discovery as a whole. So we're going to see a lot more of this in the future. At the center of this right now is AlphaFold, which in just a little over half a decade since it was released, has completely transformed 3D structure prediction. This culminates in the awarding of the Nobel Prize in Chemistry in 2024 to John M. Jumper and Demis Hassabis of Google DeepMind. And while it's largely solved the holy grail of molecular biology, finding a structure from sequence, it still has a long way to go. It's not perfect, and there are many limitations. Now, one of the key limitations to AlphaFold is that it predicts a single static structure based on the lowest energy conformation. And so we can't neglect that fact that proteins are very dynamic in nature, and many do not simply adopt one static structure. They actually shift between multiple conformations. This is very important for binding things like drug binding with small molecules, allosteric regulation, enzymatic catalysis, and folding and unfolding events as well. Furthermore, while AlphaFold does not predict the lowest energy conformation, its output also lacks thermodynamic information. That is, it doesn't provide the free energy of different conformations or how stable the overall protein is. This thermodynamic information is critical to understand things like the effects of mutations, drug binding, and drug design, as well as getting into implications involved in protein engineering. So AlphaFold also struggles with conformational flexibility. It does not provide information on very flexible regions in proteins, such as loops, hinges, intrinsically disordered regions, or the N and C termini of proteins. And many of these functional proteins will shift between conformations, such as adenylate kinase with its open and closed conformation. And so we get, we can also get cryptic pocket formation and autoinhibition, or even ligand-gated transitions. So AlphaFold will often predict disordered proteins as just poorly structured blobs, AI slop, if you will. And it gives no insights into the behavior of the protein ensemble. Also, because intrinsically disordered proteins are very common in signaling and regulation, their function depends on dynamic ensembles and not just a static structure. So, furthermore, AlphaFold does not model probability distributions. It does provide confidence metrics, such as the PLDDT score. That's the predicted local difference distance test. But it lacks a Boltzmann distribution of the sample of different conformations. So it's critical in the next phase of the development of AlphaFold to improve its ability to model the entire protein ensemble and to predict multiple conformations of protein structure. Now, fortunately, in the past week, a new algorithm was introduced. This algorithm is called BioEmu. I love that name. And this was introduced in a paper published in the journal Science that was published by a team of researchers led by Frank Noy that are working with Microsoft Research AI for Science. And now this paper is behind a subscription paywall, so if you don't subscribe to Science, there is a non-peer-reviewed version available on bioRxiv. I'm not sure exactly comparing the two. I've mostly been reading the free version on bioRxiv, which is pretty good, and it's a pretty good description of the overall methods in the paper. So if you don't subscribe to Science, you don't like paywalls, go get it on bioRxiv. I'll put a link in the description. Now, if you look at the abstract of this paper, this is how they describe BioEmu. And I'm just going to read the abstract here. So, BioEmu is a deep learning system that emulates protein equilibrium ensembles by generating 10,000 statistically independent structures per hour in a single GPU. That's 10,000 times faster than MD. BioEmu integrates over 200 milliseconds of molecular dynamics MD simulations, static structures, and experimental protein stabilities using novel training algorithms. It captures diverse functional motions, including cryptic pocket formation, local unfolding, and domain rearrangements, and predicts relative free energies with 1 kcal per mole accuracy compared to millisecond scale MD and experimental data. BioEmu provides mechanistic insights by jointly modeling structural ensembles and thermodynamic properties. This approach amortizes the cost of MD and experimental data generation, demonstrating a scalable path towards understanding and designing protein function. So BioEmu attempts to overcome many of the limitations of AlphaFold's protein ensemble prediction capabilities by generating an entire ensemble structure of protein conformations that reflect their equilibrium distribution. That is, how often each conformation is likely to occur under biological conditions. Now, it's trained on experimental delta G free energy data, as well as molecular dynamics simulations, all atom molecular dynamics. And so we can estimate relative free energies to within 1 kcal per mole accuracy. And it can predict stability changes and delta G for mutant proteins as well. So by accurately sampling alternative conformations, we can provide more accurate structural predictions of large domain motions, localized unfolding, cryptic binding site formation. Furthermore, the protein ensembles predicted by BioEmu match the known radius of gyration and secondary structure propensities, even without being explicitly trained on IDPs, or intrinsically disordered proteins. Now, what's a radius of gyration? Radius of gyration is a geometric property of an area or mass distribution, so the protein in this case, that represents how far out its mass is distributed from its axis of rotation. It's the distance from the axis where the entire mass of the object could be concentrated while maintaining the same moment of inertia. Why is this important? Well, this helps to understand how effectively an object's mass is distributed to resist rotation. A larger radius of gyration indicates a wider distribution of mass. This leads to greater resistance to rotational motion. The experimental radius of gyration of an intrinsically disordered protein can be compared to theoretical or experimental values published for a globular or an unfolded protein. So globular proteins, like all compact, unfolded is very wide, and they will have two different radii of gyration. And so this allows us to estimate whether the protein behaves as a random coil or whether it's more compact due to putative residual structure. Random coils we usually refer to highly unfolded or disordered proteins with no or almost no structural elements, with the vast majority of residues solvent exposed. And so this is critical for AlphaFold because different parts of AlphaFold will be predicted as a solid structure or kind of a random coil. So this is one metric that we can use. Now, BioEmu also explicitly learns to sample from an equilibrium distribution. And that produces structures with probabilities that match thermodynamic ensembles from long MD simulations. It's also less computationally intensive than MD simulations. And so, because it uses a scalable, efficient, generative machine learning framework that requires only GPU hours rather than thousands of GPU hours. So it's very efficient at generating its protein ensembles. The BioEmu is based on a denoising diffusion probabilistic model that's called a DDPM. And so this is very similar to the distributed Graphormer. What is the distributed Graphormer? And so distributed Graphormer, this is a type of generative deep learning model that's designed to sample equilibrium distributions of molecular structures, particularly proteins, rather than just predicting a single structure. It combines concepts from graph neural networks, that's GNNs, as well as transformers, and it's used as the foundation for BioEmu in this particular paper. In plain terms, here's how we can break it down, which might be suitable for understanding by undergraduate chemistry or physics students. And so basically it's a graph-based transformer model, and it's designed to generate full 3D structures of molecules like proteins, right? And so if we kind of look at it from a learned probability distribution perspective, i.e., it would sample structures the way nature would, right? It kind of uses what's called diffusion models as its generative backbone. Now, what is diffusion? Diffusion is pretty widely used in the machine learning community. Diffusion is a machine learning method that starts with a noisy random sample, and it gradually denoises that sample step by step to ultimately reveal the shape of the object, the protein in this case, right? Now, if you've ever used AI image generators such as DALL-E or Adobe Photoshop, they work by this same model. And so think of it kind of like a blurry photo gradually coming into focus. But the final photo is not fixed. It's just one of many possible structures that the protein can take in solution. And it's called distributional because it learns the full distribution or ensemble of all possible protein conformations, and not just one structure like AlphaFold does, right? So the Graphormer part, this is so named by combining two words, graph and transformer, right? So how does this work? Well, first of all, a protein can kind of be viewed as a graph. The nodes are different amino acids or atoms in the protein, and the edges are the bonds or the spatial relationships. And so the transformer part, the transformer is kind of like those used in language models, large language models, and this is great at modeling long-range dependencies. That is, how one part of the protein influences another, even if very far apart in the sequence. So the Graphormer is really kind of a hybrid model. It understands the chemical graphs, such as the bonding in the sequence, and then it can capture long-range spatial or sequence effects via self-attention, kind of like a transformer, right? And so BioEmu ultimately takes the protein sequence processed by the AlphaFold evoformer, right? And it uses that for its residue level representations, and then it outputs coarse-grained 3D protein structures that are sampled from a learned equilibrium distribution. Now, this coarse-grained model contains the major features of the protein structure, but it lacks things like hydrogens, water, and side chains in an effort to speed up its calculations. Coarse-grained models are really nothing new. They've been used by biophysicists and scientists for quite some time. So then once we get the model, then it uses a scoring model to actually perform structural refinement across time steps. And that scoring model is very similar to Langevin dynamics, or what we call molecular dynamics, or MD, right? Now, BioEmu was trained on a clustered version of the AlphaFold protein structure database. That's the AFDB. You can access that online. But it has augmented structural diversity, so it's got a little bit more structural diversity embedded in it. So then this training set was then fine-tuned on 200 or so milliseconds of all atom molecular dynamics, and then we've got about 750,000 experimental protein stability measurements. They call this the megascale data set. And so this was then reweighted using Markov scale models, and the experimental constraints were then better that would match equilibrium, right? So if we kind of look at the results of this, let's kind of take a look at our results. BioEmu is going to generate ensembles that include major domain motions. If we kind of look at adenylate kinase, this is a good example between the open and closed conformations of adenylate kinase. We've also generated some ensembles with local unfolding, such as RAS and P21 switch helix. And then there's also cryptic pockets, little binding pockets that are formed, such as in fasciin and aminotransferase. Now, the success for this was defined as sampling conformers within about 3 angstrom RMSD to known experimental structures. And so they looked at the root mean square deviation, and obviously a 0 angstrom RMSD means we have 2 structures identical to each other, and the higher the RMSD, the more divergent. Generally, when we're comparing biological structures this way, 3 angstroms or less is what we kind of call the gold standard, right? Now, the second major thing they observed is we're emulating MD equilibrium distributions. And so they trained and validated on fast-folding proteins. These were the fast-folding proteins that were in the paper by D.E. Shaw back in about 2011. And so that was the first paper that really demonstrated that you could use molecular dynamics to study how a protein folds from its sequence to its final structure, right? Then they also looked at the Cath domains that were greater than or equal to 100 microseconds of molecular dynamics each. And so for these equilibrium distributions, they achieved a mean absolute free energy error of about 0.73 kcal per mole for fast folders, and a little bit greater, about 0.91 kcal per mole for cath domains. So both were actually pretty good in terms of the overall equilibrium distribution. And what they also noted that this outperformed molecular dynamics by about 4 to 5 orders of magnitude in speed. And so it required only minutes to hours of GPU time versus thousands of hours on a standard, on standard molecular dynamics. So we've increased the speed to generate these computational ensembles by several orders of magnitude, right? Now we can also look at predicting protein stabilities. And so this uses that mega scale, using the experimental delta G for those predictions. And we got a mean absolute error of less than 0.8 kcal per mole with a Spearman correlation of greater than 0.65. What this enables us, this correctly identifies the identification of highly stable folded proteins from the Protherm database. And it also enables intrinsically disordered proteins with a very high radius of gyration, as well as unfolded structures. So we're able to identify all of these particular motifs right here. So that's very important. We're also able to predict mutation-induced stability changes, or a delta G with structural rationale, unlike black box free energy predictors. So overall, in conclusion, BioEmu is like the first generative model to emulate thermodynamic equilibrium ensembles with a quantitative agreement to molecular dynamics and experiment. It samples conformers thousands of times faster than MD while maintaining comparable accuracy. This makes it ideal for applications in drug discovery and allosteric site prediction, protein design, interpreting experiments. And so we're going to see a lot more of these, especially in analyzing large ensembles, because we have to remember proteins are not static structures. Proteins are living, breathing molecules that a key component of their function is their motion and how the protein actually fluctuates and equilibrates over time. That's why molecular dynamics has been so valuable in understanding protein function. Now, BioEmu is not without limitations. We're not, completely there yet, right? So right now, it's only been trained on soluble proteins at about 300 Kelvin. So we haven't taken into account membrane-bound proteins, for example. And 300 Kelvin, okay, we're not too far off from body temperatures, 310 Kelvin, but if we look at like thermophilic bacteria that might be around 360 Kelvin or 400 Kelvin, I mean, it hasn't been trained on that yet, so there's just no data, right? It also can't handle ligand-bound systems, and there's other environmental factors, things like coenzymes and small ion binding and that sort of thing. So it hasn't really been trained on some of those things yet. But unlike MD, it does not use a physics-based energy function, so it limits reweighting or rigorous enhanced sampling. So it's got a few limitations, some things to work out so far. But right now, BioEmu represents a major step forward in scalable, high-fidelity modeling of protein dynamics. And so it shifts molecular dynamic simulations from being a production tool to a data generation backbone for training fast generative emulators that are compatible with large-scale experimental and bioinformatics pipelines. So think about it this way. If MD is like filming a high-speed movie of every atom in a protein as it moves, then BioEmu is kind of like using an AI artist who's watched millions of these movies to instantly sketch millions of snapshots, many realistic snapshots of the proteins in possible poses. And so then it can tell us how often each one occurs. So we've undertaken a major step forward in understanding the conformational dynamics of proteins. This is going to help AlphaFold predictions understand how proteins actually work. And so it's going to improve our ability to properly model biological structures. And so that's going to be a fairly large leap forward, a giant leap forward in terms of biophysics and biochemistry, and as well as drug discovery. So I hope you've enjoyed this little analysis into BioEmu. And so I'll go ahead and stop here. If you have any comments to add about this episode, please leave them down in the comments. I actually read those. Those are actually very useful. And go ahead and hit the like button and subscribe to this channel. And that way, whenever I update with new podcast episodes, you'll actually get the updates on there as well. Go ahead and subscribe to my channel. I put a lot of other useful content on there. So for now, I will wish you farewell, and I will see you in my next episode, which is episode 10. So we're kind of churning right along here. It's getting pretty exciting. So thank you for watching. Thank you for watching Biochemistry Unfolded, from atoms to algorithms. If you enjoyed this content, please subscribe to this channel, and we will keep you informed of future episodes. Thank you.